Okay, so we can understand it by this diagram also. See here, this is a human cell and it has a nucleus. Inside the nucleus is the DNA. We have we used a particular restriction enzyme to cut this DNA into small fragment, or we have recognized the gene of interest and we have removed that gene of interest. We have kept it. Now what we have done here, we have taken a bacterial cell. Bacterial cell has its bacterial chromosome and a plasmid circular DNA or the plasmid. We are taking this plasmid and taking the plasmid out. We are cutting the plasmid using the same restriction enzyme that we are using to cut the human DNA. Same restriction enzyme is used to cut here also. Now we have two things ready. We have the gene of interest cut using the same restriction enzyme, and we have the plasmid DNA cut using the same DNA, same restriction enzyme. Now what we have to do? We have to combine these two. So how will we combine? We will combine these by using DNA ligase. So here ligation has taken place and a recombinant plasmid is being generated. Now this recombinant plasmid is allowed to enter into the bacterial cell again, and now this bacterial cell with R plasmid, recombinant plasmid, R plasmid, R small r is known as recombinant. So this plasmid DNA with recombinant plasmid is containing the human gene, the gene of interest. Okay, so this is a transformed cell, transformed host cell which has readily taken up the gene of interest which is being loaded in the plasmid DNA. Okay, moving forward. See, once again I have this diagram, one more diagram. This is the recombinant plasmid. See here, so many markers you can see, so many things. One can be DNA of interest, another are also responsible markers, which I will tell you why these markers are being there. So this is a recombinant plasmid in which you can see many colors. So it has many gene of interest, or it may have many different kind of genes responsible for different production, different uses, having different uses. Okay, so what will happen here? We are taking the host cell and we are taking the recombinant plasmid, allowing it to enter into the host cell so that a transformed cell is produced. So that a transformed cell is produced. Clear? So this is a transformed cell which readily takes up the recombinant plasmid. Moving forward. Now, transformation. What is transformation? What we have done, understand it from this diagram also. Then we will take up one step at a time. Today I am telling you all the whole process of genetic engineering will be discussed today in a small capsule. Then after that we will be taking isolation one day, then probably restriction enzyme one day. We will go topic wise because it's a lengthy unit. So today I'll be telling you an overview of the whole chapter, and then we will consider one topic every time. So let us start here. What we have done, this is the recombinant plasmid. Recombinant plasmid is introduced into the bacterial cell. This is the transformed cell now. Now the transformed cell is allowed to grow in a medium, culture medium, and these are growing. See, you can see the colonies over here. These colonies will have the DNA which is transformed, the DNA which is recombinant. Okay, so when these plant cells are allowed to grow, they will give rise to a new transformed plant. Okay, let's move forward. Now here you can see another example. I'll give you another example of human growth hormone production, how the growth hormone can be produced. See here, this is a human cell. We are taking the human cell, taking out the chromosome. This is, for example, this is the gene of interest which is responsible for human growth or production of human growth hormone. So we are taking that particular strand of DNA, that particular portion of DNA out. We have kept it. Now we are getting our carrier ready, the vector ready. This is a bacterial cell. The plasmid DNA is taken out. The plasmid is taken out. Now the plasmid is being cleaved. Cleaved means cut. Cut by using a restriction enzyme, the same restriction enzyme which was used here, EcoR1. You can see here EcoR1, small EcoR1 mentioned. This is the name of the restriction enzyme. Escherichia coli restriction enzyme strain 1. I will tell you the nomenclature also. So the same EcoR1 is used here and here. Okay. Then here are the protruding ends. We will combine it and this will just clip on with the gene of interest to form a recombinant DNA. See, you can see a recombinant DNA. Now this recombinant DNA is again inserted into the bacterial cell, and now the bacterial cell is having the plasmid DNA which has the human growth hormone. Okay, clear? So which kind of cell this cell is called as? What is the name given of this particular cell? This particular cell, what is the name given to this particular cell? This is the transformed cell. Okay. So here we complete today's class. This is all for today. I will be sharing with you certain important points which you will read on, and just take up your book on page number 41 and just read it. Only page number 42, 43, 44, 45 is just a table, until 46, isolation and purification of DNA. Study, just read till those points, and we will be continuing in the next class. I'll be sharing the notes for this chapter as soon as I can arrange. Okay. Thank you so much.